So because I don't want to take any time away from our presentation tonight, I will just start by introducing Danielle Garneau. She is an associate professor of environmental science and a terrific colleague and friend. And I'll let her talk more about about herself, introduce herself. And tonight our topic is microplastics in the Champlain watershed. Go ahead, take it away, Danielle.
Thank you, Elizabeth. And thanks everyone for joining in tonight. I'm going to share my screen. And that was exciting. That reminded me of my childhood. We called it the booper, and the same thing happened when we grew up. So that was pretty awesome to hear. I just wanted to share with you all some of the research that students and I have been working on since about 2012. And first preface that I am a terrestrial ecologist. My PhD work was up in Alaska working with bear and moose and predator-prey interactions. And from then I moved about the Northeast teaching in colleges, gaining experience before landing here in Plattsburgh. And my research is broad. You know, I do roadkill surveys. I'm working on the microplastics, which was totally new to me, getting into the aquatics realm. And I'm doing a lot of game camera work up at the Burn in Altona that colleagues and students and I have been working on the last couple of years. So there's a lot of different directions, a lot of interest in microplastics, especially because we're also tied to the lake. And so I want to share with you that work. And I apologize if there's a lot of pictures. And so, you know, I can't begin the study without, you know, sharing with you all the faces of all the hard workers that were involved in this project. So all of these pictured were undergraduates at the time, and I switch over here a couple of graduate students, and we've been able to embed this research into even our general ecology lab. So people not interested in doing research and moving on to graduate school or careers in science still get an opportunity to learn about this really important question. You're seeing this all right, Elizabeth?
Yes, we're great.
Okay. To introduce, I should probably, you know, discuss the trends in plastics. So you can see in this graph that we're looking at millitons. There's just been an increase across, you know, the globe in use of plastics. And, you know, the trend is just moving in this almost exponential rate. In fact, our plastics are now being sent back to us from many of the countries which normally received it. Where does all that plastic go? It goes to the landfill, a little bit gets recycled, but a lot is often unaccounted for. And it comes in many different forms. It ends up in many different locations. I'm sure you've seen pictures, disturbing pictures like this. And you've probably heard of the gyres, the ocean gyres that are circulating these currents and also circulating these masses of plastic and other debris. The work that we do focuses on the tiny things. So basically the term microplastic is anything that's less than 5 millimeters in size. There are really two different types. There's the primary and the secondary microplastics. The primary microplastics are the ones that were designed to be tiny. So these are the pellets that we used to have, or the beads that we used to have in our exfoliants and our toothpastes. And then the secondary microplastics are the larger plastics that over time and in, you know, wind and solar degradation, wave action as well, will become smaller and smaller. And so the work that we do in our lab involves characterization. We characterize across all these different methods. Generally, we're looking to characterize under the microscope based on type. So are they fragments, which are pictured here? These are just random shards of larger plastics. Are they foam? So imagine your styrofoam cup in a bead form. Are they film? So that's where your bags come in, saran wrap, food packaging. Are they pellets or beads? And that's where those exfoliants come in. And then microfibers, which are really what the focus of my research moving forward. And I think everyone's research in this field is moving. We also look at color. We use sieves to separate them out by size. And it's very important also to follow up and determine, using other methods, determine what kind of plastic it is, because that's going to signify where it's going to float in the water column or whether it will sink to the bottom, and what organisms might be exposed to them. So where are they coming from again? Tire wear, boat debris, a lot of sort of the marine debris, they call it, even though we're in a freshwater system, but a lot of the boating materials, washing of textiles, think of latex paints as well. Wastewater treatment plants are a known pathway of our use of plastics, and so on and so forth. Even cosmetics can contain them. I began this research in 2012. As I mentioned, this was not my forte. My mentor was Sherry Mason. She is a professor who at the time was out at SUNY Fredonia. Her research began by surveying in tall ships the loading in the water bodies in the Great Lakes Basin. And so we sort of began by learning the techniques she was using at the wastewater facilities, and then applied them later to a lot of the work that we're doing in our own basin. And so one of the projects that we were involved in was kind of a collaboration with the Lake Champlain Research Institute. Many of you guys might have heard of this organization. Dr. Tim Myhook, who is a colleague of mine, who is a professor at SUNY Plattsburgh, he is running the show here. He's got a lot of colleagues, and you might even recognize, this is Sue Hager, who's working on watershed issues right now. What we did was we were able to tap into their long-term zooplankton surveys. You'll notice over here on the map, all of these dots represents a location where they're surveying almost on a monthly basis for many years now, off the boat, off bridges and whatnot, using plankton nets to basically look at the community of plankton in our basin. They're looking for things like spiny water flea, fishhook water flea, and other invasive species, looking for turnover and whatnot. And what some of the researchers had noticed were these floating particulate, and that's where I came on the scene. And we were able to, we call these NURDLES. These are pre-production, we think, rubber, given the work that we've done to look at their composition. And they're interesting. It's sort of a conundrum. We aren't really sure where they're coming from, but we really wanted to see if we could figure out when they arrived and where they may have arrived. So we're looking to track them spatially and temporally. So we haven't gone through all the samples, and we've sort of kind of come to a standstill in terms of the efforts we've been able to make in terms of, staff and whatnot. But in 2010 and 2011, at least the samples we went through, and I'm going to call these NURDLES, those are those pre-production materials. We didn't see any in those 2010, 2011 samples. In 2012 and 2013, we start seeing them, and you'll notice the size of these dots correlates with the volume of them that we're seeing in these different locations. And then in 2014 and 15, the samples that we were able to evaluate, we see a fewer of them, so it's pink and not red anymore, and the locations are shifting a little bit. So we kind of thought, maybe we'll find a tributary that would have high loading, and we'd have a sense maybe there's land cover or an industry along that river that could be the source of these pollutants. Additionally, we thought if they all arrived at one time and it was uniform distribution across the basin, that perhaps there was some sort of watershed-wide event, for example, like the flooding, that would have brought them all in. But the timing doesn't quite match up with the flood, and there isn't really a location. The northern high volume up there could just be the movement of the water into that direction and into these shallower bays. So we still are not yet certain, the origin of these particular NURDLES, but they're still under investigation. We're still noting in the new samples that the NURDLES are still present, even in the recent ones. So where are these microplastics deriving? There were studies that were done in the basin involving my mentor, looking at a bunch of different tributaries, and then breaking out the results by type. So you'll see, pellets and beads here. You'll see fragments shown here, foams, films, fibers, and so on. And then down here, you'll notice that they are separated out by tributaries. So this was a very intensive survey. And each of those tributaries got a land cover evaluation. So you'll notice that, In this Indiana location here, there's a lot of urban, it's very urban, less agriculture. And so you can notice here the pellets, for example, if you trace back the pellets seem to be associated with Buffalo, New York. So it's really interesting that you may be able to pinpoint, locations, sources, that sort of thing in these studies. What the big take-home messages are, and you can see in this map right here, is that higher densities of these microplastics are in Lake Erie, the more shallow lake, the more populated lake, with a lot more industry. And you'll see here again, Lake Erie versus some of the other Great Lakes, the loading of those plastics is higher. But what's interesting also to note is that tributaries, so, we're thinking about the rivers, where are they coming from? Those rivers are higher densities than we're seeing in the lake proper. And when you look at the types, as you break them out by types, there are different types represented. Fibers are very common in the tributary samples, and more diversity in those types in the main lake, and that could be in part because there are more sources of marine debris in that main lake. So the work that we've done, again, began at the wastewater treatment plants. We learned the techniques that Dr. Mason was using over at SUNY Fredonia, and my students and I we started at the Plattsburgh wastewater treatment plant. We set out a sieve with a sump pump attached to the last stage of processing in a wastewater treatment plant. And we ran it for 24 hours. We knew the flow rate of the sieve, and we then went and evaluated the plastics that were found on the sieve. We were able to then scale that amount of plastics, given the flow rate from our little pump and the volume that we got in our sieve right here, and scale it up to the amount of water that passes through the treatment plant, and get an estimate of the daily loading of plastics through the wastewater treatment plant pathway. And you'll notice it grew from the Plattsburgh plant to a couple of other plants on the Vermont side, one that Elizabeth worked really hard with her students and volunteers to sample for us, as well as St. Albans and the Burlington plants on the Vermont side, and on the New York side, Ticonderoga. This is what our surveys look like. So, 24 hours on the sieve, and you've got foams, and you've got films and fragments. And we're doing a chemical process that breaks down anything biological and leaves behind just the plastics. And I don't need you to kind of belabor the take-home messages here, but what I want you to see is that the different colors represent the different types of plastics, right? The fibers are the darker blue. The pellets, which everyone worried about from the facial washes and the toothpastes coming into the sewer, those are always fairly low. So really, one of the take-home messages is the beads were never a major contributor in plastics overall. But every plant is different, just like people, right? Every plant is a different size, services a different population of people, has a different age, a different orientation. Some of them have stormwater treatment. Others have tertiary treatment, which is often a sand filter or some kind of microfiber filter at the very last stages to reduce the amount of phosphorus and other pollutants that can enter into the lake. So each one is different, and each of those plants ended up telling us a different story. And here's Elizabeth's plant in Vergennes. What was interesting is they actually had the least particulate that was being emitted, but the majority of them were fibers. So here's an example from 24 hours at the St. Albans plant, and it was chock full of a bunch of foam, which I thought was kind of interesting. But the big take-home message here, again, here are all the plants that we surveyed, right? We've got the flow rate from the plants, and this is what we were able to extrapolate out as the amount of particles per day that are being emitted through the wastewater treatment plant pathways. And this is just a handful of plants represented around our basin. Imagine the contributions that could be, what if we were to consider all of the plants in our area. And as I mentioned, the beads are not the full story, but what is exciting is that they got banned as a result of a lot of the research that was going on during that time. And the films are not the whole story. But what's exciting about living in New York and other states is that they are on the radar and they are moving towards the ban, right? Like we have now. What I want to focus the rest of the talk on is where I think most of the microplastic research is moving, and it's towards fibers. I'm going to show a handful of studies to just get you thinking about where the focus of these researchers are going. This is a study in a remote mountain catchment. So basically, they went to the Pyrenees, and it really shouldn't be a polluted area. And they looked at the amount of plastics that they found in the snow. And you can see all these microfibers. This is a recent, I think, October 7th article from folks at the Rosalia Project based out of Vermont. And they've been doing a lot of research on microfibers, developing something that you can place in your washing machine to catch some of the fibers and reduce the loading into the sewer. They are interested in looking at what the impact is from the lint that's going out of the filters off your house. So your dryer filters. And so they actually collected the lint after going and washing and drying this fleece blanket. You'll see all the particulate fiber and noticing from the vent, the loads are much higher, close to, but they also are found, in the yard, right? A lot of this gets picked up in the wind. And now we have a major concern about atmospheric deposition. Why is that important for us to consider? Because for the most part, most of the research has been, concerned about human consumption of these particulate. The fact that we might eat mussels, shellfish, we could, eat, fish that have retained these particulate, and then we get exposed in that way. But there are studies out there that suggest the amount of food that we intake pales in comparison to the amount that we might actually be inhaling in the dust in our own homes, which horrifies me because I haven't dusted in a long time. But you can see here, this study that, that speaks to that. And there's a lot of concern that there can be, lung damage, inflammation, right? There's a lot of physiological responses when your lungs are exposed to fibers. And so scientists are really moving in this direction, right? Any human studies, human trials and whatnot, monitoring takes a very long time. So a lot of this research is being done on smaller organisms in the lab kind of setting. But a lot of environmental toxicologists are concerned about what this exposure means for us. A lot of doctors and scientists are getting on board saying we need to really think about our food packaging. So many items are packaged in plastic that could, leach into those products. There are people that are out there in the media going and allowing themselves to be tested. For example, this was a recent study. I think he's an NGO director, and he made sure that he spent a couple of weeks eating a lot of very processed foods that were wrapped in this kind of plastic. And then they evaluated his fecal samples. So, when he went to the restroom, they evaluated that and were able to find higher loading in the week where he ate more of the plastic. So the plastic is everywhere, right? There are studies that Dr. Mason, my mentor, did where they surveyed bottled water and found that 93% of the bottled water had microplastic contamination, most of it tiny and most of it polypropylene, right? And remember, I mentioned what kind of plastic it is actually is important to know because that would indicate what organisms might get exposed to it. She and other researchers also evaluated sea salt, beer, and tap water, and you'll notice estimated, yearly consumption that can, be in the order of magnitude that you might want to, consider and reconsider what it is that you're consuming. This one really hit home. I don't know about you all, but we're in this, we're surrounded by farms, farm communities. Many of us are CSA members. And I've got young kids that, I struggle to get them to eat any kind of good food. So apples and carrots are usually the win. And this study is actually suggesting that apples and carrots of the vegetable group have higher microplastic loading as compared to some of the others. Now, they do say that it's much less than what you get by drinking bottled water. But again, something we hadn't looked about, and all of these, research is just coming out looking at everything at this point, right? We need to learn more about it. My research moved from the wastewater treatment plant into the organisms. And again, it was based on a lot of the studies coming out that bivalves and things that were consuming fish, seafood, mussels and oysters, they all have these microfibers. And so we were able to work with basin partners and go around during tournament season and get some of the fish donations, do dissections. We also looked at cormorants and some invertebrates. And these are all representative species that we evaluated, 15 species. And we were able to dissect these organisms and evaluate their microplastic loading in their digestive tracts. What you'll see here is a graph of mean microplastics per individual. Cormorants, the top predators, right? These are the birds that eat the fish, had the highest abundance of microplastics. And again, look at the color. The color corresponds to fibers, right? I told you this is really a story of fibers. Moving on to the fish, on average about 5.6 per fish. Most of them, again, fibers. And then when we look at the small organisms that the fish are eating, even less load. But again, the big story here is what is being retained in those organisms in our lake are fibers. And if we broke it out by the fish species, for those of you who are fishers, you'll notice in red, we've kind of grouped the top predator fish. In the middle, these are the ones that kind of have a versatile diet. Down here, these are the more herbivorous ones that are going to eat sort of the plants and algae. We'd expect their loading of plastics to be less than those top predators, and that's exactly what we see. And again, the light blue, the focus is the fiber. When we look at those small microorganisms, right, those macroinvertebrates, we looked at a whole bunch of them, and they all have different feeding categories, just like the fish do. And we did something similar. Some are collectors, some are scrapers, herbivores, filter feeders, and predators. Those filter feeders, similar to the mussels, had high loads, and the predators also had high loads. Again, fibers being the big take-home message. And there's just some pictures. We took each one of those fibers in the organisms, and we put them on this machine. It's called a Fourier transform infrared device. It's up in the chemistry department, and my awesome student, Erin Ashline, spent hours and days and years actually getting a read. So, UV light kind of excites the particulate through that's kind of pinned down right here. And gives us kind of an EKG readout that matches up to a library of known plastics. We were able to determine that about 1,500 particles from these 20 species, that most of those fibers and whatnot are showing up as polyester, like polyester terephthalate, but also some rayon and some nylon. And that's really important when you're doing this research, again, is to figure out what kind of plastic it is, so you know what organisms it might affect. This is all very important. It's driven, this research, not just mine. There's so many other people who are doing this research kind of on a full-time basis. This is just one of my projects. What we're learning all the time is that microfibers are on the rise. They come from a bunch of different locations. And I don't want to sort of be a downer, just like the good news we had with the ban on the beads and a ban on the bags, the films. There's now momentum building on the fibers. So they're listening out there, which is pretty exciting. There's actually policy that's been drawn in Connecticut and California and New York to add additional labels to the inside of our clothing. Here's the one from New York. This is the actual bill. That says any product that has greater than 50% synthetic material will have a label on it, that mentions something like this is a major category of plastic pollution in water and can pose, a serious threat to the environment, found within fish and shellfish consumed by humans. So I think that's pretty exciting. And there's a lot of, some of them even local manufacturers of outerwear and whatnot and gear that are very environmentally conscious and are looking for solutions. So there's a study out where, again, the Rosalia project, the people that did the work with the recent work with the lint coming out of the dryers, this is the Cora ball that I mentioned. It's a little kind of rubber. It looks like a little coral. It actually looks like a bunch of bubble wands kind of fused together into this sphere. And then you throw it in your washing machine, and it's meant to kind of act like a coral polyp would and catch the fibers. And then you can remove those fibers afterwards. This is the lint lever and after wash filter. It goes right on the end of, your pipes. That needs to be cleaned out every once in a while. And then this is from Patagonia. And you're supposed to throw your clothing in there and then wash it in there, and it catches the fibers. So there's a lot of, people thinking about what we can do, what our role might be to reduce this particular loading. And then also, as I mentioned, those manufacturers are looking to design smarter products that don't wear as much and last longer. So what are some of the things that we can do? We can certainly think about our consumer habits, right? So we can shift away what we buy, reusing, recycling. If you're in need of a new washing machine, using a front-loading washing machine rather than a top-loading has actually been shown in studies to reduce, have a reduced sloughing off of those fibers, and also the type of detergent you use. So if you can use liquid, it's better than the powder. You could consider some of those new products that might help in reducing the loading. Bringing our own bags. Well, we obviously now have to do that in our state, which is exciting. Bringing your own bottles and so on. Balloons and utensils, things we need to consider. Straws, so many straws used every day. Staying away from that as well. To-go containers, something we forget about, right? And then bottle and bag laws and so on. So there are some things that we can do. And before kind of I end and wrap things up, I just wanted to show you sort of what we've been doing more recently. So that was research done a couple of years ago, and more recently we're looking at other alternatives to counting, because it's a lot of hours under the scope these students are spending. And there's actually a stain, it's called Nile red, that will bind to plastics and will fluoresce with a UV light. And so students are starting to learn that technique. Or at least before the shutdown, we were kind of piloting that technique, and there's automation out there that will help in quantifying that, and that would save some time. We've also been interested in considering other pathways of these microplastics. I was intrigued by the amount of riverine plastic that study revealed, and I thought perhaps land cover and runoff might be one of the sources. There are commercially available, even in our local area, fertilizers that derive from wastewater treatment plant processed effluent. They've been treated with lime and other means and heat and so on to reduce contaminants, drop out some of the contaminants. But one of the things that doesn't get picked up, it's not really on the radar, are the plastics that might still be in that material. And so we, as you're thinking, it's a very, user-friendly and environmentally friendly way to fertilize the lawn. There actually might be microplastics, and we're seeing, there are microplastics in those products. And then we're kind of dabbling in terrestrial loading of microplastics. There's a couple studies that show worms, earthworms, picking up microplastics as they're burrowing through the soil. And many of you, as you're planting in your gardens, may put some plastic weed fabric down, or it kind of feels like it might be plastic, but if you read the label, it actually is derived from some type of plastic. As that wears over time, that's going to get smaller and smaller, get into the soil, and perhaps continue leaching. How does that impact plant growth? And so there was a study done looking at wheat. So we decided we would look at corn, and students were in the lab, and they had control plants. They had plants that were treated with amendment of microplastics from that weed fabric, and macroplastics, bigger size ones. They also looked at the amendment that we were talking about, and then half of them they added worms to. to see if there would be any impact. So here's our students kind of working in the greenhouse. Again, we had to shut this down a little quicker because of COVID. And you can see, you know, these are all the treatments that we had going. All the ones on the right-hand side are taller, and those were the ones that had the worms. So that's kind of no surprise. The graphs here, nothing you need to spend a lot of time on. Again, we're looking at heights and weights and so on, but really the big take-home messages were, we never saw any impact, at least from the time that the plants were grown, in the plastic treatments versus the controls. We did see enhanced growth when that soil amendment wasn't used, even though we know that the soil amendment could be a pathway of those microplastics. And growth for the plants, both weight and height, was increased when we had worms, which again is nothing new. But I'm curious to know whether those worms actually uptook any of the plastics. And with that, I want to thank, you know, funders in the past, the Lake Champlain Sea Grant, and the grant that actually got me started in this project, which was an innovative instructional technology grant, which I think is just something that I hope in the future, you know, we can revisit in some way, using other SUNY schools to make matches with our students to grow student research opportunities. And that's what happened in my situation. That's what brought me from working in a terrestrial system to working in an aquatic system. My students made a match with a faculty member at SUNY Fredonia, and I learned an entirely new technique that took me on a trajectory I never would have thought I would have done, and the students have been with me ever since. Other helpers we need to thank, obviously, all the students you saw pictured earlier, LCRI staff who went and took samples, gathered fish for me as well, and all their tireless work out doing their sampling. Elizabeth, who has spent so much time on this project and really just, this is a project of passion for her, because she spent so much time on the lake and grew up here. Getting to the bottom of this plastic pollution problem and seeing what we can all do to kind of make it a healthier lake, and learning as much as we possibly can at all the time. So I would love for you all to go back and start these conversations, to let your neighbors and friends and family know that this is just something that we should start putting on our radar, and gain some momentum with. Thank you for listening.
Thank you so much, Danielle. So interesting, so many ways. Before you put your slides away, though, we do have a few questions.
Sure.
Before we go to the questions, I'm going to just, because you got through your material so quickly, I want to just take 1 minute to add to a project that I am hoping to get off the ground. One of the things that came to my attention right away when I started working at the Maritime Museum 5 years ago is when I was teaching students how to sample for plankton. Every single plankton tow we got these blue flecks of blue flecks. I didn't know what they were. I assumed they were plastic. And so after some time, I was able to get enough samples and compare. I collected the samples from the plankton tows as well as samples of blue products around our campus, sent them to Danielle, and she identified them using that same process that she was using for the water treatment samples, and found that they came from the blue plastic barrels that are used for flotation under docks. So that really made me uncomfortable. And to where we've gone with that is that we are now trying to, we are replacing our docks at this time and trying to find flotation that is less, that degrades less. The blue plastic barrels that many private landowners and commercial marinas use right now it's a blue barrel that's designed for food. So it's a food grade plastic. There's not really good information about its usable lifespan or its safe usable lifespan in the conditions that we have in Lake Champlain, and particularly the UV exposure that it's not designed for. So we are looking for partners right now to try and design a study to evaluate other types of flotation. The other type of flotation that's typically used is sort of these big cubes of plastic encased styrofoam blocks, which when that external skin degrades, that styrofoam is all exposed to the lake and definitely appearing around the lake. So that is another source of trouble for the lake. I know this information is all pretty, it's pretty overwhelming. And I will say that in my work as an educator, I try and be optimistic because it was humans that thought about this amazing product that's light and strong and colorful and so useful in so many ways. And I really do address young scientists and young students interested in science as this is a puzzle for us to solve. And chemistry created it, and chemistry can probably solve it if we put our resources into this. And I believe that's true. So that challenge is out there for students every day. And for those of you who may not have really realized how important those chemistry lessons are, that's the direction that I'm headed as an educator, is to help students really understand how to find solutions. So Danielle's work quantifying and qualifying and describing the plastic problem is the first step towards getting to some great solutions, which we hope can emerge from what we're trying to do. That's my little talk about what we're doing, but I would really like to get to some of these questions. So Danielle, if you will, keep sharing your screen and we might need to refer back to some of them.
Okay.
I'm going to go start at some of the ones that were, let's see. Well, a question from Mary Nell. Have there been studies of humans of the effect of consumed microplastics?
They are just beginning to trickle in. Like, I think that's why I was taken by that more recent one that I took the screen snip on. And it's a sample size of one. That's the thing. There are very small sample sizes. This one in particular, again, I think he was, it was maybe a month, and he did a couple weeks in a low plastic exposure diet and a high plastic exposure diet. I think you're all kind of science minded, so you can imagine, he's catching samples in the restroom and sending it off to kind of undergo a process similar to what it is that we do, and just getting a sense that the loading was substantially different in those two different trials. But that's a sample size of one. And the only other direct study like that, there was only one direct study like that I've seen. But like I said, in order to be able to have a high sample size and be able to evaluate this stuff with statistical significance, you have to bring it into the lab. And in many cases, that begins with the smaller organisms and then works its way up. I know there's been a lot of discussions and even in the literature a little bit about how small can we get. So, if you were to take a blood sample or other fluid sample, could we get nanoparticles of these plastics? And I think there have been a couple of studies looking at that. I'm not that strong in the chemistry angle, but I did share it with some of our biochemists who have interest in it, that there have been suggestions that these particulates, imagine a fiber, imagine that fiber is coated with some kind of polymer that is so, tiny that over time that could easily get into the lymphatic system, the blood system, and circulate. So they're beginning these sorts of things, evaluating what those impacts are on the human body.
The other thing that we talk about when we're talking about the consumption or ingestion is whether it stays in the gut and the length of time that it stays in the gut.
Yeah.
So there are a lot of variables in that. And the other thing is there is this study that Danielle, it used to be in your presentation. I don't think it's there in this presentation, about the study, the effects on fish in a tank after ingesting plastic and the change in, so the polymers are the plastics in general mimic endocrine and neurological hormones. So that's the effect that was seen in the fish population. Is that correct, Danielle?
Yeah. So that study was interesting. It was kind of a food chain study. They actually treated like the algae with the nanoparticulate, and then the algae was consumed by, say, like the Daphnia or the copepods, so those macroinvertebrates. Then the macroinvertebrates were consumed by the fish, and they observed their schooling behavior, a bunch of untreated versus, treated fish, and their schooling behavior changed completely. So there was some neurological effects that were observed as a result of that. Yeah.
Thank you.
And Elizabeth makes a really good point. This is a snapshot. That fish that we dissect, that is a snapshot of their digestive tract. So I'm sure that there is particulate that enters and exits, and maybe we caught, it was just in that period of time. But what I'm thinking, the reason why in part we're seeing a lot of the fibers and not some of the other particulate in those samples is because, think about what happens to fiber. Think about like your beater bar on your vacuum, right? All those fibers, my hair, the dog's hair, it's all tangled up. And then it like enmeshes in the digestive tract epithelium. And then it's stuck there, right? And over time, it may slough off. Over time, it may degrade and get uptaken into the system that way. But I think that's why we see more of those fibers, is the nature of the fiber.
Thank you. So Greg asked the question, in the time frame of 2012, you mentioned that I think this refers back to the NURDLES, when you mentioned that they were not there from flooding, but Hurricane Irene occurred in the fall of 2011.
Right.
That didn't seem to line up.
Right. So we don't think it's associated with the flood, is what I was saying. We were hoping that we would maybe see that it was timed directly with those samples. That, for example, it may be the waters rose up and overtopped, say, railroad beds, because I've given this talk before and people have said, train cars carry a lot of those pellets and NURDLES, and, the lake is surrounded by, rail beds, and it makes sense that that could potentially be where it comes from, because there's oftentimes some spills and whatnot. But the timing of the events, the intense storms and the spring snow melt and the timing that caused that mega flood, it just didn't seem to align with when we began seeing it in the samples. And this is early, right? There are so many cabinets of those samples, and we had those students working hard for a summer, but we haven't been able to go through all those samples. So, we have an N equals maybe 100 or 200 bottles, but there are thousands. So maybe we just didn't get to the rest of the story.
Greg also has a question. Did you notice any differences in the wastewater treatment plants, whether they use anaerobic, aerobic, or fixed film digesters? Is there any correlation to the plastic output?
That is a really good question. And I probably would have to go back and look at the components of those systems. I have a table that basically speaks to the fact that all of them are treating stormwater, and or all but one, I think, are treating stormwater. And I think two of them, St. Albans and Virgins, have tertiary treatment, but I'm not quite sure about the digesters. But that is a really good question. I can go back and reach out to them to find that out.
Thanks. Catherine Reinhardt asked the question, can you repeat that about apples and carrots? Do they hold microplastics or not?
Yeah, let me go back to that so that you could also follow up with that article. That was a fairly new one in the last iteration of my study, of my talk, that I wanted to pull in. Here it is. Are you guys still able to see?
Yep.
Okay. They're up to this point, and this was in August, there had been no microplastic data available for vegetables and fruits. That's how new this is moving, right? There was, up until I want to say a year or so ago, there was limited, if any, studies that showed that it was airborne and found in these remote areas. There was one study that I recall from Paris, where they had, you know, dry particle collectors on the tops of roofs, but that was the first. And so here, you know, microplastics less than 10 microns, depending on the type of vegetable samples. Apples and lettuce were the most and the least contaminated, so you're better off eating lettuce than apples, which I don't know about that. Estimated daily intake, and this is where like my kids come into play, right? Estimated daily intake of adults and children for apples, right? You know, in my mind, you know, kids eat a lot of apples, especially during certain seasons. But over here, I wanted to snip this from down, you know, later in the, in the study. This thing always gets in the way. Let me see if I can move it. Yeah. So again, therefore, although the abundant presence of the microplastics in the investigated vegetables was of considerable concern, exposure to the microplastics through ingestion of these foods was less than that of the polyethylene terephthalate bottled mineral water that we drink. So it's something to consider, just like all the new things as we do more research we're learning about, but the loading is not as great as that, as if you were drinking bottled water every day, which I know a lot of people actually do.
Yes. Another question from Greg. When a boat is registered, or is there any literature provided to them regarding their contribution to the problem? I can pretty well answer that.
Yeah, go for it.
No. So there are so many pieces. If you were to inventory this, and I can say this with confidence because I've done it, if you start your day when you get up out of bed and you count how many pieces of plastic that you are in contact with in the first hour, I was in contact with 60 pieces of plastic within the first 15 minutes that I did that in my own home. When you look at boat parts, you start to see that everything that we use, whether it's cars or boats or kitchen appliances or the toothpaste top and the light switch and your eyeglasses, and it just goes on and on. It's so prevalent. It would be hard to kind of point directly to what the contribution is from any one boat, especially because so many parts are made of plastic, and they are made of so many different kinds of plastic and manufactured in so many different ways and subject to so many different mechanical influences, never mind UV influences. It's just really, really hard to characterize the contribution of one product. And
go ahead, Elizabeth.
I was just going to say, and I think it would be a great, great thing that for any product. So there is a movement for extended producer responsibility. So the idea of that being that if you make plastic, if you manufacture plastic, you have to be the one to figure out how to repurpose it or dispose of it. And that is not a very popular idea among manufacturers because it's so expensive. And so, but that is a topic for a future conversation, what a cyclical economy might look like if the manufacturers were responsible for the disposal or repurposing of plastic.
Yeah. What I was going to say along those same lines, I think having an informational pamphlet, some sort of campaign that you could kind of pair with the cleaning of the boats, you know, as you're, you know, trying to reduce the aquatic invasives, would be really, really useful. It reminded me, and I don't fish, but it reminded me of someone who asked me at one point in a talk about lures, plastic lures. And that is an entirely, you know, and fishing line, that's an entirely other, you know, source there that, you know, just being aware of that loading.
Mm-hmm. And I will say that it's very, very, very hard to just strictly eliminate plastic from your world. So even if you have a CSA, you put everything in brown bags or in cardboard boxes, or just every little act that you take in your life to try and reduce plastics, getting to plastic zero is going to take a very, very, very long time. It's just too practical of a problem, of a product, and a material. But there still may be some answers we don't know yet. So let's take another question. Stephen asks a question: could you review the effect of these microplastics on human health? So we've sort of somewhat answered that, but you want to summarize again, Danielle?
Well, I think it is multifaceted, and you know, there's not you know, a ton of studies out there. This is just, you know, one study here that compared muscle intake and microplastic inhalation from household so the dust, right, when you're sitting down for a meal, you know, fibers entering into your bronchioles, you know, so causing inflammation in your lungs, which could, you know, over time result in fibrosis and granulomas. So, you know, like sort of reminded me as I read this, like this is sort of what miners and asbestos, you know, folks that are exposed to this might experience, right? Severe tissue damage when inhaled over time. So these studies, and I haven't read them, I should say, you know, maybe speak a little bit deeper to that. Whether or not they challenged the cells in a cell culture rather than in the human body, you know, as I mentioned, a lot of this work has to happen in the lab, and the sample size needs to be high. So I can imagine a lot of this derives from kind of cell culture work done in these labs. But fibers persisting in the lungs, you know, depending on the length and diameter and so on, you know, and you can see this visual here of what the process could be.
We did provide a list to the Lyceum committee with some of these studies, this references. So if you would like more information about any of these studies, give us contact us, send us an email or what have you, and we'll get some more information from Danielle to be able to, if you'd like to pursue any of this reading. It's pretty dense reading, so we didn't know that everybody would want to go into each study, but we'll be glad to track those down for you.
If you're really interested in this topic, the journals to keep an eye on are Environmental Pollution, Marine Pollution Bulletin. Those are some of the big ones. That's where a lot of the plastic research is going. The more recent one on the fibers came from PLOS 1, PLOS 1.
We have two more questions, or a question and a comment. Why is liquid laundry detergent better when it comes to plastic? Powder can be bought in cardboard containers.
Right. So that is a very, very good point. And that may balance it all out. That's probably, I don't know if that's something that they even factored into the study, and kind of unfortunate. Yeah, I'm thinking of borax, you know, good old fashioned borax versus, you know, Tide or something along those lines that's in the container. I don't know. It must have something to do with the abrasive properties. I know the situation with the top loading versus front loading is the number of revolutions and the speed at which it can flip the material, and it you know, abrases against each other. And so, you know, considering that, but you know, having talked with some of the folks that are working in these manufacturing facilities about the types of products, they're looking at these smart weaves, these smart blends, you know, like, do we need, you know, tighter weaves so that, you know, the material doesn't fray as often? Is there not only the tighter weave, a new weave that we haven't kind of considered, maybe kind of looking to nature and doing kind of looking at biomimicry to see if there's something out in nature that seems to kind of resist and be resilient, like the hold fasts that you find on the, you know, the mussels that are sticking on rocks and stuff. Is there some sort of new material that can be derived from something like that that would be more resilient up against it. So, you know, there's a lot of R&D going on to try and reduce that. They are definitely aware.
Thanks. Again, Catherine Reinhardt comments that she has a core ball and it does work. It doesn't capture as much microfiber as she thought it would, but still it's some's better than none, I guess. I would agree with. Thanks, Catherine.
Yes. And I'm looking back to the one slide where I have the picture of the core ball. I don't know if I have the associated graph. I don't, but the study right here that evaluated, if you go on and Google Scholar and type in lint lover and Coraball, it will show up. There's a study that you know, has like control versus lint lover versus Coraball, and the amount of fibers that were in the wash water as a result of that. Coraball was effective. I think it was statistically significant, and lint lover was more effective than all. But the difference in cost, again, just to kind of, I'm sorry about this panel being in the way. The cost differential, I think this is about $20 for one, and this was, you know, 150 to 200. And, you know, I had actually considered maybe doing a study in a dorm at the school with the lint lover, to kind of get a, you know, increase awareness with the students so that they could envision how much they're contributing to this pollution problem. But then I learned that these need to be changed, you know, every 3 or so times. And I assumed that was going to cause an overflow in the dorms and not be effective at all.
I think another topic that we've talked about in the past, Danielle, with another audience has been applying pressure to manufacturers. It was like, you know, the emissions controls on cars are expensive to add to the product design and manufacture. But likewise, if these filters were a requirement for washing machines, that might be a big way to control the input. And if I remember correctly, there was a comparison or a study that said, I think it was up to 3,500 fibers per garment, between 1,800 and 3,500 garments per fiber per wash.
Yeah.
What for fleece. And I will say that in defense of some of the innovators in recycling, Patagonia, the Yvon Chouinard, who started Patagonia, thought that he had solved a big problem by putting plastic recycling bottles into fabric and making fleece, and put a lot of R&D effort into using that to solve a problem, too much plastic. And was, if I understand correctly, and this is, I only heard this, I don't know if it's really true, but that he was so distressed at the study that showed how many fibers were emitted for each washing of a fleece garment. He commissioned his own study. And when his own study kind of corroborated the results, he basically said, stop the train, we have to refigure this out. We have to come up with new technology, a new manufacturing process, a new something that we have not solved this problem. And I don't know that made it worse, but the problem isn't solved yet.
Yeah. And it's interesting because, you know, I've given these talks so long, and like I said, this is just one of my avenues of research. There are people that have devoted their entire life to, you know, tracking down these microfibers and looking at alternatives. I'm really surprised at the slower response that I would have expected it to be faster from the washing machine and the dryer manufacturers. To, you know, build something into their design in these new models. I mean, we have a fairly new one. I don't see anything but the regular lint filter, which, you know, I have to say that new study that I put in here, I haven't even read it, everyone, but I'm definitely going to when I get back. You know, we kind of assume that that's all the lint and we're going to remove it and all is well. And I hadn't even thought about additional that's leaving through the vent. And causing that airborne particulate, adding to that airborne particulate that we were talking about. I am just really surprised because it seems like that is definitely something that could help. And I'm not sure why it hasn't happened yet.
So we're getting, oops, it looks like we're a little past 8. So we have 2 more questions, or 1 more question, 1 more comment. Marianelle asked the question, what are some of the efforts to remove plastics, microplastics from bodies of water?
Yeah, for the most part, they haven't been entirely successful. There have been some vessels that have been deployed out into areas near the garbage patches and, you know, like booms, and then having some sort of a conveyor belt to kind of draw it in like vacuum style. And then, you know, unload it back elsewhere. But there's always, or at least, you know, I see it, you know, there's efforts to kind of do this, and then a couple weeks later it breaks down and then it has to be repaired. And I don't think that's necessarily going to be the big solution. There's also been, you know, investigations on, is there some sort of a bacteria or, you know, bioremediation kind of component here. And interestingly enough, for any of you that have maybe have apiaries, there was a study that was brought on by somebody cleaning out their apiary and putting like residual stuff into, say, like a Hannaford plastic bag, or, you know, one of the plastic bags we can't use from grocery stores anymore. And they forgot about it and went back a couple weeks later, and there were holes in the bag. And it turns out that the worms that were in the hives that they were removing, I don't know that the worms had survived, but the bacteria in the guts of the worms had survived, and it's that organism that was digesting the plastic. So there have been studies on these particular organisms associated. I think they're called wax worms, and we actually did some trials in our lab, a student research project, Alexandria Elliott. So we've got a poster out there, and she, you know, put these worms in ball jars. You can actually buy them to feed to your lizards at like PetSmart. I thought these must be some exotic things, and then I called, and you can actually get them locally. But you know, she exposed them to different types of plastic, and it was really, you know, if you think of the plastic that you put your oranges and your produce in, how it's a little bit more flimsy plastic than maybe some of the other bags that we're familiar with. That one was the one that I think we saw the most, you know, marks on. But we didn't have any bacterial culture. It was more the organisms themselves that we exposed them to. So I think, you know, and there's also a really cute, I think it's more for an educational purpose. It's called Mr. Trash Wheel. I encourage you all to Google it, because we all need something to lift us up these days. Imagine a big snail with googly eyes that sits at the mouth of your river and does sort of what that, you know, boom would do. It's like pulling in the plastic as it's coming through. And I have talked to folks downtown affiliated with the wastewater treatment plant and Public Works, saying that I'd really like to get us a Mr. Trash Wheel down at the mouth of the Saranac.
Well, in fact, when you, again, in an earlier presentation, I remember you talking about one of the wastewater treatment plants, I think it was Plattsburgh, that had a super, super high daily count of microplastics in their output. And they did one simple thing, which—
Oh, yes. Thank you. Yeah, because that's a really good thing to point out. So with every upgrade that a plant makes, being able to go back and evaluate the impact, we saw a dramatic decline from one like season to the next, or you know, we had winter, so normally we don't survey in the winter, and we went back, you know, the next season, and the drop was like dramatic, like orders of magnitude in the loading. And I immediately called them up thinking something must have happened, and he said, oh yeah, we replaced our coarse grate filter, the first filter barrier before anything enters into the into the treatment plant process, that kicks back some of the bigger stuff. And they got a more fine, gradation, separator right there. So that helped explain that. So anytime a plant, and a lot of the plants in our area are older and are now getting funding to get these upgrades, anytime that happens, I think it's really important to kind of get the pre and post evaluation.
Well, it stands to reason that if you keep the big things out, then they will not break down into the tiny things that get out the other end. So that's a nice argument for the keeping the waterways and the stormwater drainage as well away. I mean, keeping the big things out of there to start with, that's flowing into the treatment plants. One more comment we have from Greg to patronize wool products over products that are more than 50% synthetic solves a lot and requires a lot less washing. Couldn't agree with you more, but sheep come with their own set of environmental impacts.
Yeah, and actually
an automatic solution.
Right. Well, and they're also, just like somebody who mentioned that thing about the, the detergents, which now I'm like, oh man, the wool thing is interesting because it really does depend, right? If you're thinking about the impacts of these fibers on the organisms that are out there intaking, or just as they're swimming and their gape is open and they're uptaking it that way, not even through like trophic transfer. What if wool fibers were sitting in a pile, were sitting in gasoline or in some other contaminant, right? And then that gets uptaken and adheres. And so, I can imagine that if it comes in contact with something, or if the wool is treated with something, and oftentimes it is, I don't know how all of the blends are, then you can imagine that it could have it could behave similarly.
So lots more things to consider. Danielle, I'm going to have you, if you don't mind, stop sharing your screen.
Sure.
And I just want to thank you so much for sharing this information. There's so much for us to learn and so much for us to figure out in life. I'm going to share my screen and just thank everyone who's attended. If you like what you saw, feel free to make a donation to the Grange. It helps us to keep doing these things. We appreciate your patience while we work out kind of the bugs in our format, and we are really open to your input and your feedback. Please send it on into the Grange. We do have some great upcoming programs, and next week we will be hearing from a really interesting program by Amy Godin about the statue of John Brown in Lake Placid, and should be very interesting. We will skip a week for the election night dinner on November 3rd. There will be no Lyceum that evening, but we will definitely be resuming for 2 more sessions. One will be about agroforestry and the other about climate change in the Adirondacks. So that will wrap up our Lyceum series. There are other movies and music events that you can check out at the grangehall.info. So with that, I'll stop sharing and say goodnight. Thank you again, all of you. It's really great to see such amazing participation from the audience and from so many attendees. Thank you so much.
Thanks for all the questions.